When you really get down to it, life on Earth is solar powered. The chloroplasts of plants use a process called photosynthesis to capture light energy from the sun and convert it to chemical energy that can then be used by other organisms. So photosynthesis nourishes almost all of the living world, either directly or indirectly. We've talked before about photoautotrophic nutrition, light-based carbon fixers, so they're able to take inorganic carbon sources using the power of light to make organic carbon. So the chloroplast is ultimately the site of that. Now, the chloroplast is not necessarily an organelle. It could be something like a bacteria that has photosynthetic capabilities, because that's where it evolved from in the first place. But for the most part, we're probably talking about chloroplasts. And you can see on the right, the chloroplasts are part of the plant structure. They're found inside of the plant, specifically in leafy plants in the mesophyll cells, or the spongy mesophyll. And inside the mesophyll cells, you will find these chloroplasts, and inside the chloroplasts, you will find the thylakoids, and inside the thylakoid space. That surface area and the compartmentalization allows for that efficiency, but it's really going to depend upon light to power the process. So light is a very wide spectrum of electromagnetic energy, but we think of chlorophyll as green because chlorophyll is not able to absorb the green light. It reflects it or transmits it. So the other colors of the light spectrum are the ones that are actually being utilized here. If you look at the process, it is a redox reaction, just like cell respiration was, with the molecules coming from CO2 and water becoming the sugar with a waste product of oxygen. Some of that oxygen will go directly to the plant's mitochondria, but it will produce more oxygen than it needs, and that excess is what we're breathing. But light and water will power the first half of photosynthesis, the so-called light reactions. Those are going to be the ones that are actually fixing energy molecules. The water is going to be split into hydrogen ions, electrons, and oxygen. The oxygen is the waste product. The hydrogen ions are used for a variety of purposes, and we have gradients of pH we can measure to show that, and the electrons are used for their reducing capabilities. So the light reactions are going to be carried out in the thylakoids, and it's going to convert light into chemical energy. It's going to use water and release oxygen. The other half, the Calvin cycle, or the carbon reactions, are going to take the CO2 in and rearrange the CO2 into sugar using the reducing power of the oxygens. So the idea that plants turn CO2 into oxygen is a misconception. That's false. Plants turn CO2 into sugar. They turn water into oxygen. When you really get down to it, though, it's really a light-powered process. Chlorophyll reflects green, so if you look at the absorption spectrum, it's going to be very, very low in the green spectrum. Now, chlorophyll is not the only pigment that you can find in plants. There are a variety of them. There are multiple types of chlorophyll even. You can see 3 examples of pigments here. There are other ones like the anthocyanins and the xanthophylls, and they have very characteristic color spectrums. But if you look at the so-called action spectrum, the actual rate of photosynthesis, which is measured by oxygen release, you will notice that it is a net of all of the pigments found in the plants. So that higher point in green shows me that there's definitely some pigments going on here that can absorb some green light, but green is definitely the least efficient in its absorption. The original experiment where they figured some of these things out was done by a man named Engelmann, and he used algae and aerobic bacteria. The aerobic bacteria use oxygen, they're aerobic, so there was more bacteria found in regions of higher action, and that's correlated to our understanding that we have now of how this works. But when you get down to it, the chlorophyll is a pigment, and any pigment is a molecule that's able to absorb light. By absorbing that light as photon energy, the electrons are excited. They go to a higher energy state. They go to a higher level, and when they fall back down, the difference can be used to power a process. If you just isolate chlorophyll and have it release its energy within an isolated system, it's going to fluoresce this sort of pinkish color because it's releasing all of those colors at the same time. So it's kind of like the color circle has converged on that magenta section. But for the most part, the pigments are going to be capturing those electrons, capturing that energy, and passing those electrons to a special pair of chlorophyll molecules in a system called a photosystem, which is a group of proteins and pigments that focus this reaction. That is going to mean that these electrons, instead of falling and wasting the energy, the falling will be used to power a process. We can see what that looks like in the next slide. So there are actually two main systems, the photosystem 1 and 2. They are named in order of discovery, not in function, so they're actually backwards in naming. Photosystem 2 is going to be the first one. Photosystem 2 is going to absorb light, primarily in the spectrum of 680 nanometers, which is why the chlorophyll is here called P680. That will then pass electrons to an acceptor, which will be linked to a series of proteins in an electron transport chain. The chlorophyll will lose oxygen, so we will have to split water to get some electrons back, not oxygen. That does mean that it has to be more electronegative than the oxygen, which is unusual, but it is where the oxygen actually comes from. That's powered by light and it's called photolysis. The electrons, though, are passed through the electron transport chain through the cytochrome complex. And the cytochrome complex is a proton pump, which will power ATP production, which we'll look at in more detail in a bit. The second photosystem, which is actually photosystem 1, uses a slightly different spectrum of light, P700. You can see that here. And that will then excite the electrons, which will then be used to power an NADP plus reductase. So this is going to be our proton carrier, our electron carrier, picking up, going from NADP plus to NADPH. Now, plants don't need as much NADPH as they do ATP, so every now and then this linear process, or this Z path, is redirected into a circular process. It's called cyclic electron flow. That means that the electrons from photosystem 1 can be used to power the cytochrome complex to produce more protons, to produce a greater ATP production. So there are options in the later path. But basically it's kind of like this here, that it's as things move up, they can fall down. It creates potential energy that can be harnessed. The proton pump from photosystem 2 is then going to, between photosystem 1 and 2, is going to then create a proton gradient, which we can measure by looking at the pH. That proton gradient will create a proton motive force, allowing the flow of the protons through ATP synthase, just like we saw in cell respiration. That proton motive force will allow the production of ATP. Photosystems 1's NADP plus reductase will power NADPH, and we'll now have all of the energy that we need to power the Calvin cycle. NADPH is a modified dinucleotide, just like NADH and FADH2, but this one has an extra phosphate group attached, so that's where the P comes from. But I always thought it was helpful to remember that the electron carrier with P is the one that is in photosynthesis. Notice that I didn't say the one that's in plants, because plants have mitochondria as well. They're going to have all three of the electron carriers, where we're only going to have two of them. The Calvin cycle, or the carbon reactions, or the Calvin-Benson cycle, the Calvin-Benson-Hussein cycle, these are all names for the same system. They are going to be what actually turns CO2 into sugar. This depends upon the enzyme called rubisco, or ribulose bisphosphate carboxylase oxidase. This is our carbon fixer. So the carbon fixation process is going to take the 5-carbon RUBP. It's going to put that into the rubisco enzyme, which will then attach the CO2 molecule, making a 6-carbon. Now, you're going to have 3 rubiscos working at the same time, allowing it to fix it, because only every 3 CO2s will release a stable product. But each rubisco is only really able to take in one at a time. So this picture is a summary. The 3 CO2s will join to the RUBPs to make a 6-carbon sugar, which is completely unstable and will break into a 3-carbon sugar. So now I went from 3 5-carbons plus 1 3-carbons to make 3 6-carbons. Now I have 6 3-carbons. Those are then going to have some ATP applied to rearrange into this bisphosphoglycerate, which you don't need to remember, but the ATP is applied to rearrange to a more stable structure. And then NADPH will be applied to utilize that reducing power. So in phase 2, the reduction is going to create a stable and reduced high energy sugar called G3P. One of those G3Ps will be released for every 3 CO2s that enter, 3 in, 3 out. The remaining 5 3-carbons will then need to be rearranged or regenerated using ATP back into the 3 5-carbon RUBPs so that we can maintain the cycle. But it is a circular, cyclical process that continues again and again and again. Now, the Calvin cycle is incredibly old. And it evolved when there wasn't a lot of free oxygen in the atmosphere. So it's ribulose bisphosphate carboxylase oxidase, which means that it's able to take in CO2 or O2 pretty much equally. It can't tell them apart. So when plants close their guard cells, they're going to have a buildup of oxygen inside their cells and a loss of CO2. Why would they close their guard cells then? Well, for water retention. So sometimes Rubisco can get confused, and instead of pulling CO2 in and powering the Calvin cycle, they instead bring O2 in and power a photorespiration cycle. So instead of building sugars up, they break sugars down, and they're going to release CO2 and ammonia and a variety of other chemicals. This is going to be energetically negative and can be harmful for the plant, but it's sort of a holdover ancestrally. And this is one of the major roles of the peroxisome in plants, is to neutralize these negative impacts that come from this photorespiration process. It will mean that there is a loss of energy overall, but the maintenance of water levels can sort of balance it out. Plants have evolved some ways to deal with this. So to limit the negatives, they have evolved two other major pathways of energy capture. Normal photosynthesis, or so-called C3 photosynthesis, is called C3 because it releases a stable 3-carbon sugar. For C4 photosynthesis, this is going to be a spatial separation. That means I have two cells. Instead of having my Calvin cycle occur in the mesophyll, it's occurring in another cell, the bundle sheath. So the CO2 is going to come in and it's going to be fixed as an organic acid. It's going to be fixed by an enzyme called PEP carboxylase. That is a carboxylase, not an oxidase, so it's not going to get confused. This 4-carbon organic acid will move across into the bundle sheath, where it will be converted into CO2 and utilized in a standard Calvin cycle process. But because the O2 will be released in areas outside of where the Rubisco is active, photorespiration is drastically reduced. So C4 photosynthesis is a spatial separation using PEP carboxylase. Now, the same enzyme can also be utilized in the so-called CAM photosynthesis, or crucillation acid metabolism. This occurs in a group of organisms called the crucillations, like pineapple or Spanish moss, which are actually pretty closely related. The CO2 will be fixed by PEP carboxylase at night, and that will then fix as the organic acid. There is no spatial separation. This is all occurring in the mesophyll, but the CO2 will be utilized by the Calvin cycle during the daytime, and that will then lead to a reduced utilization of oxygen by Rubisco. Both of these things are going to have some inefficiencies because of the energy transfer, but it's less efficiency issues overall than if they were to rely on standard photorespiration processes. This is more common in areas that have a high allow of water loss, like tropical environments or desert environments, but we can see that there are still changes to occur and evolution that's occurring within plants. So who knows, maybe there'll be new types later, but that's where we are now as far as the evolution of plants, and I hope that made sense.